K Gupta, a clinical trial of botanical drugs. Dr. Gupta.
Senior experts with me here, and I'm sure that the issue which I'm going to talk here is complex, is a lot of questions, a lot of unsolved issues, but I'm sure that with the thought process which will come into this, particularly when Professor Kazi, Dr. Handa and Dr. Rai Chaudhary is sitting here, I think we will have solution coming out of it. When we talk of issues in botanical, particularly clinical trial, I think is not as simple as what you do with NCEs. The issue is primarily because on one hand we have years, maybe decades or maybe centuries experience of safety, so-called perceived safety or established safety, versus when we talk of NCE, which is absolutely new molecule, which is entered into the body, we do not know how it's going to behave. Should they be treated as same at par, or there should be some bridging study, or should they be treated differently as far as the toxicity testing is concerned and the clinical trial design is concerned? I think these are the several issues which will come here. We have been talking on the why the drug development, particularly in botanicals, particularly in India and for that matter in this region, has not given the desired outcome. If you just see that how much money and how much projects and how many scientists have been working on these areas, but what is the product outcome? I think we have miserably failed. And I think that this is the time when we have to really do introspection to see that is there a need to have an alternate strategy? Is there a need to have an alternate thinking process? Or is there a need to have a different clinical trial pathway than the established what is as per regulations of NCEs? So that is what I will just flag some of the issues. Many of the issues have been raised again and again, but I will just put some of these issues here. If you just see what are the traditional medicine challenges today, is primarily when you talk of a clinical trial, that means the first important thing is that you have to have investigational product. And the quality of the criteria of the investigational product which you should have has to have a uniformity, a standardized, and it should have a stability, and it should be same at center A and center B. It should be same as you are doing clinical trial, versus it should efficacy will not be changed if variability exists. And that has been the major limitation of the clinical trials, forecasting into the drug development and marketing sustainability which is concerned. That is what is the first major things. And large emphasis has been given, and Professor Handa and Dr. Kazi will agree with that, that's how much effort is being done to say that yes, these are the criteria for standardization, this is the HPTLC profile, and this is the stability data is mandatory before you put this. And large number of, you just see, even the WHO has reported the large number of clinical trials have no validity because the investigational product was not standardized, and therefore the entire effort, entire data goes into waste. Another issue is the lack of specific research methodology. If you just review, we have reviewed large number of clinical trials, particularly on herbal or botanicals. We get mixed up. When we do a bridging study, we do not have a strategy that what type of bridging study is required, whether we have to completely follow the NCE mode or we have to completely follow the Ayurvedic mode, and then you mix up and then somebody challenges that you have not done RCT or you have not taken adequate number of samples, or this investigation was not done, this biomarker was not studied, and therefore it is very important that you have to have a standardized protocol which is acceptable not only to Ayurvedic physician, not only the botanicals, but it has to be acceptable as per the modern scientific technology. And that is what has been lacking, particularly when we reviewed the scientific protocol which has been developed by AIIMS. And here there is a need that you shed off your inhibition, your ego, your sentiments, your emotions, and come to a platform which is then exposed to scientific scrutiny, and that's what important thing is. This is the inherent issue, particularly when you're talking of a multi-fractions, multi-component herbal extract, which it may have 1 to 100 of the molecules, and for which you do not know that which is the active, which may be the highest peak, which may not be active pharmacologically, but it may be active, it may be acting synergistically, it may be acting antagonistically, and that's what the conventionally what we phrase we say that you go on fractionation, it may lose activity, and therefore you have to decide at what stage you have to stop. Go, no-go decision has to be taken, that whether we are interested in doing clinical research, a clinical trial of a single molecule or 1 or 2 molecules, or at what stage we have to stop fractionation. It's not necessary, particularly when talking of botanicals, that you go on extracting single molecule. That's one way, but not necessarily always. So you have to really do a very, very strategic thinking process that yes, in this condition, we will not go beyond isolation or beyond extraction or subfractionation or whatever it is. And then where the sufficient reason, sufficient evidence is involved. So that is you have to have a dynamic correlation, and that dynamic correlation will depend on what is the evidence you are generating. This is what you call as a limited scattered clinical data. If you just see today, whether it is for anti-diabetic, so most of the trials are anti-diabetic, anti-inflammatory, and some trials have also been started for anti-cancer drugs. These are very scattered. And if you just see hardly any trial you will see which will fit into the scrutiny of the what you call an adequate sample size, adequate biomarker study, adequate investigational biochemical parameters which have been taken, and therefore every article has a some or other deficiency, and that's why it is important that you develop some parameters which will give you the idea of efficacy. If somebody is talking of anti-diabetic and has done only blood sugar and not taken the glycosylated hemoglobin parameter, I think, and not taken the other parameters of dyslipidemia effect on, then I think is half done. The same thing could have been done at the time when you are developing a protocol for that, and therefore the efficacy in terms of when you say diabetes is not enough that you do blood sugar. If it's not enough that you do HbA1c, it is important that you also have some parameters to assess the outcome. And that is what important thing is, that when you consider the protocol development is. And similarly, when you say the absence of structured pharmacovigilance, which is also true for even the modern or what you call the contemporary medicines, we do not have, and therefore if anything which is likely to be toxic, which is likely to be causing a side effect, or which is likely to cause a drug interaction, we are missing that. And what we do not have a pharmacovigilance activity, therefore this will go unnoticed till you notice it, and when you notice it, it will be too late, and then it will be not a science, it will be in what you say a social behavior when somebody reports it. And therefore it's important to have a pharmacovigilance activity in this. It's a long way to go it, because pharmacovigilance in a single molecule is easy, but when you are doing a pharmacovigilance in a complex, when you talk of botanicals, you do not know whether which of this component is causing this side effect, and therefore the causality assessment is much more complex, and this is again an important science. When you talk of regulatory control, I think we have a very rigid as far as the regulatory control, particularly NCE or the modern drug is concerned. You have a new drug definition which is for 4 years, and you have to submit PSUR every 6 months, and then next 2 years you have to submit PSUR every 2 years, and then you have lot of issues where it is mandatory and it is obligatory and it is responsibility of the doctor and the patient to report and to take note of it. However, if you just see the botanicals, these things are inadequate, if I say so. And that is what important thing is in the clinical trials of that. Now, if I just see that this issue was flagged all over the world, that how the regulations can be put into place for the clinical trial, for the introduction or for regulation of the botanicals or for the herbals drugs. And you see, this awareness is going on, and as per WHO, 110 countries have placed some regulations as far as the botanicals introducing into the market is concerned, whether FSSI or whether Schedule Y or whether Schedule X, whatever it is. The important thing is that none of these 110 countries have any harmonization. We have a harmonization as far as Schedule Y is concerned, GCP is concerned, but if you just look into the regulations, particularly on the WHO has given a zero draft, but all countries have highly differential regulatory system. And therefore, if you are doing a clinical trial in India, it may not match what is requirement of the clinical trial in US or Japan or European countries. And therefore, this is also important. When you do a clinical trial, at least you see that how much you are covering these areas, and so that when you are introducing a drug, not only in your country, in other part of the world, then you do not come as an isolated person that you have to do this, you have not done this. And that is what important consideration when you plan a protocol of that. Now this is categories. I think we will not go into the detail of this. This is a broad categories which has been described as for herbal products as per Indian GCP guidelines is concerned. And this, when we debated about 4-5 years back, that how we can take into consideration the existing experience of safety and efficacy versus a new compound. And that's why we put it into 3 categories, categories 1, category 2 and category 3. Category 1 means used for indication mentioned in ancient literature. That means if a drug is mentioned for a particular purpose and you are using for the same purpose, no indication change, no component change, no method variation of preparation. So process is same, component is same, indication is same, then it comes as category 1, and then here we say that the phase 1 is not mandatory. But important thing is that animal toxicity is still to convince not yourself but convince the scientific world that this is safe, that you have to have a bridging study 4 to 6 weeks toxicity and in 2 animal species. Now here, whether 2 animal species is not been identified, whether it has to be rat or mouse, or it has to be rat or monkey or whatever it is, I think this creates sometimes confusion, and this creates a dilemma when you are producing this data outside the world. And this is category 2 is the new indication. If it is identified for hypertension or if it is identified diabetes and you are using for another purpose, then I think the indication changes. And once the indication is changed or the dose is changed or the composition is changed, that is what it is, the novel product, then it is considered as good or as bad as a new drug. And therefore, the entire gamut of toxicity testing is required. Now here, I would say there has to be debate on this, that if you are extending if this is just an extension of the indication, but indication has changed, if this is an extension of your dose, but the dose remains within the safety limit, then do you require this all things? I think this is the gray area. If I was giving 100 milligram dose for a particular thing, and now I am giving 50 milligram, the dose has changed. The duration has changed because I was giving 3 times a day, now I was giving 4 times a day, or the dose was given 3 times a day, now I am giving 5 times a day, and so on and so forth. As per definition, the requirement remains, this is as category 2 and 3. And this requires, I think, again a manthan, and this requires to be dealt with so that the process of bringing product from botanicals is scientifically justified and also safe. Now when you say guidelines for ASU clinical trials, be carried out only when a competent Ayurvedic, Siddha, Unani physician is also an investigator. That's what the regulation says. If it is being done at All India Institute of Medical Sciences or done at Medanta Hospital, if the drug belongs to any of these categories, it is mandatory that the person belonging to that category should also be investigator. However, when person belonging to this category is involved as an investigator, he is important that he also knows about the drug and how to conduct a trial. If he doesn't know, then it is as bad as not having him. And that is what the unfortunate situation most of the time is. And therefore, it is important to create human resources in all the system which will understand the science and which will understand the statistical basic concept, and therefore they are really actually working partner in a clinical trial, not as a sleeping partner or not just to comply with the regulatory requirement that yes, I have an Ayurvedic person in my trial and therefore I meet the requirement. That is what happening today. The issues is the most clinical trial. This is of Ayurvedic approaches have been small because the sample size was not calculated before the trial was started, because sample size calculation requires a pre-assessment of the difference in the therapeutic outcome which you expect, and that will determine the power of a study and that will determine the number of sample size, which is exercise has not been done in most of the places. And that's what ultimately when you come out with the result, you say that this trial has been done, but the trial does not fall into the category of meta-analysis, which can be taken into, and therefore you exclude it from meta-analysis. And therefore hardly any meta-analysis on the clinical trials on Ayush product is available. Had problems of research design, that's what I am saying, whether it has to be done crossover, it has to be done double blind, it has to be single blind, and again they have an issue. Lack appropriate control groups, which we'll talk just now, and therefore scientific evidence for the effectiveness of Ayurvedic practices varies. This variability of the same product, this variability because of the same product but it done at different places is highly variable because the design is different, because the outcome parameters have been taken different, but the cutoff line has been taken different, and there is no perfect protocol. The Ayush has attempted to make a uniform protocol, but those protocols, if you just critically analyze, are still require lot of critical assessment and before you adopt them. Now, what you say evidence-based medicine, we always talk of the evidence-based medicine, evidence-based medicine, evidence-based medicine, and therefore these all things which have already evidence of hundreds years, thousands years again, we are asking for evidence again. Now this has to be very critically looked into. What we are looking at, we are reassuring the efficacy. When you say reassuring the efficacy, that means you have to give an efficacy. I say there are three labels. One is you have a perception that this herb or this botanical is effective against arthritis. This is my perception because my Dadi Mada was using it. The other is experience because my Dadi and my granddaddy and I have also used it. I experienced, my neighbor has also used it. This is an experience. Neither perception will work nor experience will work. It is what is required as an evidence. And when we talk of evidence, it has to be either statistically valid that out of 100, 70% people benefit. Of these 70% people benefit, their blood sugar reduction is 10 to 15%. And if the blood sugar is more than 300, it doesn't work. And this, how many percent has a side effect? That is what called as statistical evidence, that you call as an evidence based on the investigation, whether radiological or biochemical or statistical. So this what evidence has to be generated for efficacy. And same when you talk at safety, when you say the conventional statement that any drug which is herbal is safe is absolutely unsafe statement. When you say anything herbal is relatively safe, must be a relatively safe statement. Because you have not assessed this scientifically, you have not assessed critically that how many patients have a possible of increased liver function test or increased or decreased kidney function test. That is also a side effect. They have not been assessed. And therefore, you are always liable to be criticized that no, you have not tested it, and therefore, if any drug causes any side effect, it is blamed that the drug or the formulation or entire gamete is toxic, whereas the toxicity may be because of the ingredient, maybe because of the faulty manufacturing process, because of the adulteration, or because of the wrong method of preparation, which is different from that has been described in what you say conventional Ayurvedic literature or Unani literature. That's what the major problem here is. The quality is a major issue here. As compared to standard rigid quality control, so-called rigid quality control. Of course, I am not talking of the Ranbaxy getting and Woodard getting warnings like that, but still there is a reasonable quality control mechanism exists. But if you say in the Ayurvedic preparation of the botanicals in the market, though they are supposed to be in GCP, GMP certified labs, but the large number of this is not certified. And the result is the publication in some journals which say that all the drugs coming from India have high metals and therefore they are very toxic, which is again a toxic statement, which is not true perhaps. But then one fish like this creates entire pond smelling. That's what the important thing here is. And therefore the quality issue is a very, very important issue. And particularly when you're doing a clinical trial, you have to ensure that at least your investigational product has no question of quality on that. Efficacy versus effectiveness. This is a major concern which I feel, particularly when you talk of the different system of medicine. When we talk of contemporary medicine, when you talk of anti-diabetic or anti-hypertensive or anti-cancer, you are treating a target. You are approaching a target. And therefore you are treating the symptom. Whereas in other part of the system, you are not treating the symptom. You are treating the body as a whole. So you are not looking at the efficacy, perhaps you are also looking effectiveness, which is broader aspect of efficacy. And that's why you also have to consider the quality of life, the ease, the longevity, and the economics, and how the person behaves. This is the composite of everything. That's what the difference when you do the clinical trial here is. And that's why you come out with the negative results of the clinical trial, but though the drug may be effective in literature when you analyze that. Keep in mind, absence of proof. You found that this drug is not effective, is not proof of absence. It's not proof of absence. Therefore, you have to see if a person is using this drug for 100 years and finding it effective, and you have not found it effective. Find out the reason. If there could be another mechanism, if there could be another philosophy, if there could be another effectiveness system, what you say, not an efficacy, but effectiveness. And that's what important thing is, you incorporate in the design these aspects before you design the trial. When we talk of researching Ayush, is a long-standing history of 5,000 BC, and I don't know whether this 5,000 BC should be called as somewhere it is 4,000 BC, but the highest is the 5,000 BC. Now, what we talk of, why GCP now? Why GCP now? As if everything was not good earlier. It was good, but now here is the validation and harmonized Ayush. The need of the day, when you talk of Indo-US collaboration, you are penetrating into the European or American market. At least you have to match as much as possible, harmonize with that. And that's what you have to see, that how much we can align it, what we call evidence-based medicine. And that is what important thing is. And that is what also will increase the credibility in global village. Now it is called a global village because today I think you can go and you can spread your drug any part of the world, provided you can say that yes, this is efficacy and this is safe, which is most important. And promote holistic medicine. So the refined application of Ayush with improved understanding of evidence. If you have an evidence that yes, I am having this hepatoprotective drug, not the most common because it has been used by millions of people in India and in Russia, and therefore this